fragment. That's the part of the molecule that binds to an antigen. It's the two arms of the molecule. And notice that the arms have a constant region and a variable region. Now, these constant regions within a class of antibodies like IgG are the same. Whereas the variable region is going to be made up of amino acids that give that region of the arm a configuration that is complementary to the antigenic fragment that it binds. Think of it kind of as a lock and key type of situation, where the makeup of the variable region is going to give that area a three-dimensional shape that's going to be complementary to whatever it binds to. Okay, so that's the region that binds to the antigen. Okay. Notice that there's a complement binding site. When you take pathway, that will need some. And there's a binding site down here for other cells, such as macrophages. So that when an antigen, it can be either bacteria, it could be antigenic cells, when they're coated by antibody, then this binding site has an affinity for macrophages. So macrophages will hook on at this site and phagocytose whatever is coated by the antibody. Okay, so the coating by antibody increases the affinity for macrophages. It makes whatever is being coated by the antibody more attractive, if you will, to macrophages. The term we use for this is opsonization. We say that the antibodies opsonize. Whatever those antibodies are bound to. Or we can say that the antibodies serve as opsonins.
Repeat the definition of that again.
An opsonin is just simply a molecule that, when it attaches to a foreign body, it makes it more attractive to macrophages. And it increases phagocytosis of that particular substrate.
Thank you.
I'll give you a corollary in terms of human disease. There's a condition called ITP. Have y'all ever heard of ITP?
Yeah, and idiopathic.
Yeah, and the old name is really idiopathic thrombocytopenic purpura. They've kind of changed it to immune thrombocytopenic purpura. It's the same disease. It's an autoimmune disease where in children, the child produces antibodies against their own platelets. So as a result of this immune system going rogue, now the antibodies circulate coated with antibodies. Well, when those platelets then travel through the spleen, what happens? All of a sudden the macrophages are attracted to the antibody coating and they phagocytose the platelets. So the patient's platelet count plummets because the platelets are opsonized. See how I did that? Did a little analogy there. I'm good that way. Yeah, special. Sarah, you talking? You're all smug there with your bow on your head. She went with the bow today in the Florida.
Oh.
Yeah.
Wait, why y'all smoke?
Oh, yeah.
Yeah, you just look smug there.
I'm just concentrating.
It's Friday, so
All right.
concentrate.
IgA is a class of antibodies secreted onto the surface of mucous membranes. IgA looks more like this, by the way. It's a dimer. meaning it consists of two monomers linked by J chain. Okay? And so this provides surface protection along these membranes. If any bacteria try to penetrate the mucous membrane, they get coated by IgA, and then as they invade our body, they'll get taken out.
That's a dimer?
A dimer, because it's made up of two monomers. Okay. Look here, IgM looks like this. This is the one that's the largest. It's what's called a pentamer. So IgM's claim to fame is it's the largest of all of the immunoglobulins.
Is the IgE the one that does the allergic?
Mm-hmm. Mm-hmm. So IgM, is it going to cross the placenta? Why not?
Because it's too big.
It's too darn big. It's too darn big. Now, let you know another secret about IgM. When B cells first respond to an antigenic challenge and differentiate into plasma cells, the first class of antibody produced by plasma cells is IgM. So for a few days, that IgM, that plasma cell is producing just IgM. Okay? And then after a few days, it undergoes class switching. It'll switch to IgG. So the first antibodies produced in the setting of an acute, particularly bacterial infection, is IgM. I'll give you an example of where that's helpful. Have you ever heard of mycoplasma? Anybody? Mycoplasma?
No.
Have y'all taken micro?
Oh, yeah. Good.
Okay, mycoplasma is an atypical bacteria. It doesn't have your typical cell wall. You know how in micro you learn about cell walls? Humans don't have cell walls, but bacteria do.
Right.
And they're made out of a substance called peptidoglycan. So Sarah, Chris,
Yes.
y'all will take that right now.
Yes.
Well, mycoplasma is an atypical bug. It doesn't have all that stuff. So that as a result, you can't gram stain it. You know, anytime somebody comes in, let's say with pneumonia, or if they come in with a urinary tract infection or an abscess that we're draining, the first thing we're going to do is collect some of that infected material and gram stain it. And so right away, we're going to differentiate it into either gram positives or gram negatives, and we're going to differentiate its form. Is it a staph? Is it a strep? You know, we're going to help further define what type of infection this is so we can initiate antibiotic therapy.
Right.
And at the same time, we're also going to culture it so that 2 days later, when we get final culture results with sensitivities, we can fine-tune the antibiotics. That's the way we do it. Okay? That's the way it's done. Well, mycoplasma, because it doesn't have a typical cell wall, it can't be gram stained. Secondly, it can't be cultured. Okay? So it makes it more difficult to identify a mycoplasma infection. Now, what kind of infection does it typically cause? It causes pneumonia. So it's called mycoplasma pneumonia. And it tends to cause pneumonia more in young and maybe middle-aged people. It is a milder form of pneumonia than your typical pneumococcal pneumonia, which is produced by Streptococcus pneumonia. Okay, so how do we identify it though? Well, because we can't gram stain it, because we can't culture it, we have to do serum titers. Serum antibody titers to mycoplasma. Okay. If it's an acute mycoplasma infection, then the lab will report out elevated IgM titers to mycoplasma. Now, the fact that they're IgM tells me it's an acute infection. because those type of antibodies are the type that are produced immediately upon response. Now, if instead the lab reports elevated titers of IgG, it tells me nothing. It may mean that that person had a mycoplasma infection 2 years ago. Okay? But if it's IgM, it tells me it's an acute infection. Okay? So that's where that information, for instance, could be useful. I like micro. I like the lab. You know, you just, but you realize when you work in a lab, you have bacteria swirling all around you. Huh?
Yeah.
Now I think of it as building my immune system.
Did you get sick a lot when you took the lab?
Huh?
Did you get sick a lot, or did you take?
No, I
No,
didn't.
it's not.
No.
Is it like, but it's like inactive forms of.
No. What your deal? But in the lab, most of the time you're working with non-pathogenic bacteria. Now, if you are
Not
working
doing
with
like
pathogenic
anthrax.
bacteria, you work under the hood. You know how labs have a hood?
Yeah.
Okay, so they have an updraft that pulls any bacteria up instead of letting them fall on you. Okay, and you use proper technique. Okay, so IgM biggest, and it's the first produced. Okay, IgE. Okay, this is, as Jordan says, we think allergies and parasites. Allergies and parasites. An allergic reaction is a hypersensitivity reaction to some type of environmental protein. So people who have allergic reactions are hypersensitive to something in the environment that the rest of us are fine with. Okay? And so let's suppose someone is allergic to ragweed. Now, the exposure to ragweed is mostly going to be through inhaled air. And the first surfaces that inhaled air comes into contact with is the nasal mucosa. Okay. Now, when we inhale air that's laden with ragweed, let's say during the spring, then we pick up some of that ragweed antigen. In this case, we'd call it an allergen. Okay, and because we're sensitive to it, our immune system's going to respond. We're going to develop a humoral immune response where we produce antibodies. Well, with allergic reactions, those antibodies are of the IgE type. Okay, and then with the IgE, it's going to come to, and by the way, that first exposure to the allergen is not going to result in a clinical response. Okay, it's just going to sensitize our immune system to that particular allergen. The IgE that's formed is now going to coat the surface, for instance, of mast cells. You remember mast cells that are living in the nasal mucosa. And by the way, IgE is a monomer, just like IgG. Okay. So these mast cells are chock full of these inflammatory mediators. Okay. And the IgE is now sitting on top of the mast cells. So that in the future, if we come into contact with that allergen again, likely through inhaled air, then the allergen is going to bind to the antigen binding forms of the immunoglobulin, like this. And that binding of the allergen to the IgE is going to trigger degranulation. These inflammatory mediators. Now, one type of inflammatory mediator is histamine. I think we've talked about this. Histamine is going to cause blood vessels to dilate. And certainly along capillaries, it's going to cause the endothelial cells to contract. Well, that contraction opens up these intercellular clefts and increases extravasation of fluid, so your nasal mucosa becomes congested. Okay, so your nasal mucosa becomes red, flushed with blood because of the vasodilatation. It becomes congested. And the histamine can actually cause itching when it binds to nerve endings and triggers sneezing. Okay, some of these inflammatory mediators are eosinophilic chemotactic factors. What do they do? Well, they simply draw in eosinophils from the bloodstream into that tissue that's been exposed to the allergen. So now in that tissue we have lots of eosinophils. We talked about this, didn't we, when we talked about blood?
Mm-hmm.
And so if you look at the tissue that's involved in an allergic inflammatory response, you see lots of eosinophils. You see it in eosinophils. That's what tells you it's allergic. Of course, then, these eosinophils are going to help modulate that allergic response. For instance, one of the things that the eosinophils do is release histaminases, which are going to begin to break down the histamine so that reaction doesn't go on forever. So the eosinophils will modulate that reaction. But the idea here, by the way, get back to the is that IgE is involved in allergic. So that if someone has a particularly bad food allergen, I'll go further. If someone has an allergic diathesis, a propensity toward allergies, and this is common where you have a patient who's got allergic rhinitis, what people call hay fever, allergic asthma, allergic conjunctivitis, they have eczema, they have all kinds of allergic conditions. You'll notice that their IgE titers in their blood are high, because that's who they are. Okay? They're what we call atopic individuals. Okay? Does it mean they're a bad person? Sometimes. But it just means really that they're atopic or they're allergic. All right. So let's look at this. This is a response curve of our immune system to exposure to an antigen. Okay, this is antibody titer here. This is first exposure to an antigen. We'll call this time 0. That's what people like to call this time 0. And it takes about 10 to 12 days for our immune system to ramp up and generate the immune response. Okay. During that time, what's happening is, if this is bacteria, they're proliferating. They're entering into lymphatics. They may be carried to regional